Dear colleagues, it's a pleasure for me and for Czech Glaucoma Society to have the possibility to talk during your online meeting. My name is Marek Fichtl. I am head of Glaucoma Center at General University Hospital in Prague, Czech Republic. I have prepared for today a lecture regarding the genetic basis of primary open-angle glaucoma. Primary open-angle glaucoma is a multifactorial disease where significant influence of genetic factors. In the majority of cases, there is an interaction between genetic and non-genetic factors which together leads to dysfunction of the trabecular meshwork and structural changes of retinal nerve fibers and ganglion cells. In these situations, the individual genes have a limited ability to cause glaucoma, and the pathology requires a combination in several genes and the interaction with the environmental factors. We can speak of primary open-angle glaucoma as a complex disease of inheritance. However, there are genes that play a key role in the pathogenesis of glaucoma, and their mutations lead to the dysfunction of trabecular meshwork. Even the role of environmental factor is really important. Mutation in these genes can be identified as a primary cause of glaucoma. Only few genes are strongly associated with the pathogenesis of primary open-angle glaucoma. Absolutely the most known gene is myosilin. Then we can find genes called optineurin or tank-binding kinase 1 that are specifically associated with normotension glaucoma. Mutation in myosilin cause up to 5% of all primary open-angle glaucoma cases and 1/3 of juvenile open-angle glaucoma cases. In most cases, the inheritary is autosomally dominant. Autosomally recessive mutations could be associated with primary congenital glaucoma. More than 70 pathogenetic myosilin mutations have been described yet, and it differs according to the severity of the case and age of manifestation. These myosilin-associated forms of glaucoma are often manifesting before 40 years of age and are refractory often to medical therapy. The main hypothesis of the pathological mechanism consists of the apoptosis and loss of cells in the trabecular meshwork, which will lead to trabecular meshwork dysfunction and intraocular pressure elevation. Mutations in optineurin gene reduce the resistance of ganglion cell to external factors, including the intraocular pressure. Mutations in this gene are associated with normotension glaucoma. Optineurin protein protects the retinal ganglion cells from apoptosis. Optineurin also performs this protective function in other tissues, and mutations in these genes also occur in other neurodegenerative diseases such as amyotrophic lateral sclerosis. Sometimes mutations lead to increased production of the protein instead of this reduction. Another gene associated with normotension glaucoma is called TBK1. If present in large amounts, it causes the disease by deactivating the healthy protein optineurin. TBK1 gene is coding for the protein tank-binding kinase 1, and together with mutations of the optineurin gene, it is estimated to cause about 3% of all normotension glaucoma cases. We can find a growing list of genes with some association with glaucoma. Data are available from large population studies. These studies compare large group of glaucoma patients with healthy population. The results show which mutations are more common in the POAG patients. Reported genes have only limited association with glaucoma, but there is a clinically significant genetic predisposition with the influence of the environment that would lead to manifestation of primary open-angle glaucoma. During the last 10 years, we have followed two families with primary open-angle glaucoma and autosomal dominant inheritance at our department. We can see the data from two family trees here. There are two unrelated families in which two different mutations of the myosilin gene were found, and this was the first time The specific myosin mutation was confirmed by genetic testing in Czech Republic. Here we can see the clinical findings. There are significant glaucomatous changes and visual field changes in older patients from our group. Patients from the second family, which are younger than 45 years, have normal visual field. The diagnosis was made prior to development of visual field changes. Even surgical approach was needed in a majority of cases. There we can see results of RNFL measurement, which corresponds to the excavation, optic nerve, and visual field changes. In the first family, we can see really high glaucomatous changes and visual field deterioration in 2 cases of the older patients. In conclusion, I would like to summarize what I consider to be the most important information. Primary open-angle glaucoma is a multifactorial disease in which a significant influence of genetic factors is important. Forms of primary open-angle glaucoma with early manifestation and familial occurrence are almost always with autosomal dominant inheritance. In most cases, there are mutations of the myosin gene. These mutations lead to hypertensive glaucoma. On the other hand, mutations of other genes are associated with normotension glaucoma. This mutation leads to reduction of resistance of ganglion cells to external factors, including intraocular pressure. These mutations are rarer than the myosin mutations. Most often, primary open-angle glaucoma inheritance is complex, and it is not possible to identify the primary causal gene mutation. Juvenile open-angle glaucoma is often associated with various myosin mutations. Identification of specific mutation and of carriers of the mutation in families makes it possible to individually set the regularity of prevention, of preventive care, monitoring, and early recognition of glaucoma progression and the specific treatment. I thank you for your attention.